Hello, and welcome back to Brain and Behavior. This unit we're going to be talking about the functional units of the central nervous system and, well, the nervous system in general. We'll first talk about neurons, how we came to understanding them as the building block of the central nervous system. Talk a little bit about the role of glial cells in function, dive into the anatomy of the neuron, and talk a little bit about the genetics that are involved in producing proteins, what the cell needs to function. Talk about some genes, cells, and behavior, how that relationship exists. So again, this is chapter 3 that we'll be going through in your textbook. So you'll want to make sure that you review that material as well. And this is meant to help in that understanding. So let's get started off. Early on, there was some debate about what the fundamental unit of the central nervous system was. Raymond Cahall was a researcher that was interested in what are the units of the central nervous system, and he used microscopy and Golgi's stain technique. This Golgi stain technique basically stains, I'd say, 2 to 7% of neurons in the central nervous system, but it stains them completely. And what this lets you do is once you're looking in the microscope, you're actually able to draw the entire neuron. And this picture up here does a nice job of, is one of the drawings from Cahall, showing the amazing structural differences that exist between neurons. These structural differences suggest to Cahall that there were functional differences. Some neurons may be specialized in processing one type of information, whereas other neurons were specialized in processing other types of information. And so the structure was an important indicator of the functionality of the neuron. And so he argued for what we know now as the neural doctrine, that neurons are the fundamental building blocks of the central nervous system. Well, he borrowed the stain from Golgi, and this is one of Golgi's stains down here. And you can see a very different kind of picture from Golgi's stain. You can see that there are many units, but they're almost connected in a web or a network, and the units look very similar. And Golgi argued that these were part of a network, and that if you were to eliminate one, you would eliminate all of them from the network. It's almost like if you had a garden hose and you would poke a hole in one part of the garden hose, water would start coming out, but at the end, the water level or the speed that the water would leave the hose would go down. So by eliminating one unit in that network, you're essentially influencing the entire network's functionality. So he argued for a network. Well, this debate and discussion continued on, and in 1906, they shared the Nobel Prize for their work looking at what is the fundamental units of the central nervous system. Cahall eventually was shown to be neurons are these independent units, but it came from the debate, the active scientific inquiry that Golgi prompted by his work. And so it's kind of interesting to go to the Nobel webpage and read their acceptance speeches. You can still see at that time they both were ardent supporters of this idea of the neuron either being the fundamental unit of the central nervous system, Raymond Cahall, or Golgi's belief that it was one large network or web. And if you would take one element out, that would disrupt the rest of the network of connections. So from this work, we now appreciate that there are a surprising array of neurons, and their structure is really related to their function. First, we have sensory neurons. Sensory neurons are gathering information from the body or the environment. And we have a variety of these. Some here, for example, this is an example from the olfactory epithelium, and they're going to be gathering chemical information from the nose, and they will then transduce or turn that chemical information into a neural signal and then communicate that into the central nervous system. Other ways that this can occur at sensory neurons is looking at somatosensation. So if you press on your arm, you're going to be deflecting or causing pressure to increase in a certain part of your body, and that causes stress receptors in the processes of a sensory neuron to open up, allow certain chemical reactions to occur, and then that neuron will start firing and communicating the signal into the central nervous system. So sensory neurons are really collecting information, whether it be mechanical, chemical, or information, for example, from the electromagnetic spectrum, light, from our environment, and communicating that into the central nervous system. That's the real function of these sensory neurons. And we can see there's some differences here in their structure, depending on the type of information they're collecting. Now, interneurons serve as that interface where they can store or process information. And there's an amazing amount of variety of interneurons. So they're receiving information from these sensory neurons. And we see three different types here. First, stellate cells in the thalamus. Remember, the thalamus is one of those areas that's critically involved in getting information from the body or the cortex and communicating it on. So before any information from a sensory system can get into the cortex, it has to go through the thalamus. And we see there's a very different structure in this stellate cell than what we see in a cell that's within the cortex. Here is a pyramidal cell that's found in the cortex, and we see that it has these dendrites. Dendrites collect information. A cell body, we'll see that that's involved in processing information. And then we see there's just one axon that comes off here, this pyramidal cell, and that's carrying that highly processed information onto the next neuron. In contrast, we see another amazing kind of cell here called the Purkinje cell found in the cerebellum. I should say up here, this is a immunostain for a pyramidal cell in the cortex, and we can see they almost look like a little bit of an onion, one of those onions that have the green processes here, and there's the white bulb. So they have a very interesting kind of characteristic. The Purkinje cell, on the other hand, looks a little bit like fan coral, in that it's in one plane, there's all these dendritic branches coming off, but it's all within just one plane. So if we were to rotate this Purkinje cell and rotate it, it would become very, very flat, and then you'd see it take up a lot of space. And this is an example of a Purkinje cell processed in tissue. So the Purkinje cells show this very dramatic structure where there's a lot of information it looks like it's trying to collect from. Here it's stellate cells as well, but there's fewer areas on the pyramidal cells. So these structural differences have consequences for the function of that cell, and these interneurons are in that interface between collecting sensory information and then storing it, processing it, and sending it out to our motor neurons. And our motor neurons, this is actually a wonderful picture demonstrating the processes coming off a motor neuron and synapsing, here's a motor endplate, on muscle fibers. And so as that signal is coming down this axon to communicate or release acetylcholine, we can see right here where it's going to synapse and then cause this muscle fiber to contract. So motor neurons are that final part of the processing where information comes out to the muscles and causes them to contract. And that's the role of motor neurons. We're going to talk more about the structure of neurons a little bit later, but first let's talk about another class of cells within the nervous system, or the glial cells. glial cells are really important support structure for the central nervous system. And really when I first started in psychology and I took my first kind of brain and behavior class, there really wasn't a lot known about glial cells. And I think the most I recall them saying is, well, they're glue. They glue things together. Kind of the essence of why they're called glial cells is that they are gluing things together. But since then, there's been a dramatic increase in our understanding of their functionality in the central nervous system and the roles that they play in the recovery or repair process. And we'll talk about that in some detail. I think this is an area that's only going to grow even more in terms of understanding their contribution to the communication in the central nervous system. But we have a better understanding of how neurons function. It's just glial cells are coming along a little bit later in the story here. So we'll talk about several different types of glial cells. The first is the ependymal glial cell. And this cell is important for secreting cerebrospinal fluid. And as you might anticipate, where do we see a lot of cerebrospinal fluid? We see it in the ventricles. And so here's a picture of the ventricles here. And where this red area is located, that's the choroid plexus. That's where we find a lot of these ependymal glial cells that are secreting the cerebrospinal fluid that fills these ventricles up. And you can see that there is this flow from the lateral ventricles to the third ventricle and then down through the fourth ventricle where it's absorbed and it continues throughout the central nervous system. One of the things that can happen is if there's a blockage here in the cerebral aqueduct, what might happen is that these cells will continue to secrete cerebrospinal fluid and it won't be absorbed at the same rate. And that's where you have hydroencephalitis, or water on the brain. And it's one of some of the conditions we'll talk about over the course of this semester. This is one of those conditions that is fairly easy to treat by placing basically a straw, or what they refer to as a shunt, into the lateral ventricle. That will allow some of the pressure to decrease, and a lot of the symptoms that are global across the central nervous system, such as attention, speech, memory, motor function, those symptoms will resolve fairly quickly, and the person will be able to lead a normal life with this intervention. So again, these glial cells, the ependymal glial cells, secrete cerebrospinal fluid, and they line the ventricles for that process of secreting cerebrospinal fluid. Another important glial cell are the astrocytes, and they support the neurons. They are kind of the support staff for these neurons. They do a lot for them. In this picture here, what we see is a blood vessel. That's what this is. And you see the astrocyte has wrapped its foot around the blood vessel. And from this cartoon, you can see that it's actually have processes coming in contact with the neuron. Astrocytes actually form or feed these neurons. So they're taking nutrients from the blood vessels, the capillaries, and it then transports them and feeds the neurons from those blood vessels, those capillaries. So they form the blood-brain barrier. They not only feed them, but they also protect them from certain things that might be in the blood that shouldn't go into the central nervous system, and protect them in that way. So they form the blood-brain barrier and not allowing everything to get through. Another function of astrocytes is that if there's some type of damage that occurs in the central nervous system, let's say a stroke or something, open head injury, something where there's an acute damage, what's going to happen is the astrocytes will actually form a kind of physical barrier that will quarantine or keep those damaged neurons, those dying neurons, and their proteins and neurotransmitters in place. So it kind of sets up a work area that allow other glial cells to come in and do their job during this repair process. So astrocytes feed. They set up this blood-brain barrier to keep poisonous things out of the brain, as well as they quarantine. If there's some type of brain damage, they're trying to block that and keep it in one location or quarantine off from the rest of the brain. So very important functions that those astrocytes provide for the central nervous system. Microglial cells, on the other hand, are part of the immune system, and once there's some type of damage that occurs in the central nervous system, they actually migrate and they will then go to that area where there's damage, and they'll clean up that brain damage by trying to remove those pieces of the neuron and its internal materials that may cause additional damage later on. It tries to remove those. And so here's a picture of a microglial cell. There's an area that was damaged. You see a lot of microglial cells there. What I'd like to do is show you a little movie of microglial cells in action. And I'll also post this in Blackboard. But what we're able to do here is stain microglial cells. This is in living tissue. And this red line you see here, that is a blood vessel on the surface of the cortex. And it's showing this red because there's a certain dye there showing that. And then microglial cells, as you can see here, are green, in this green color. And as I play this video, what you'll see is a little yellow square exposed. That's where they're going to shine a laser that's going to cause some brain damage or a stroke. And what we'll see is how the microglial cells respond to that stroke. So I'm going to play the video, and so you see there's normal, the microglial cells are kind of moving around. There was the stroke, and now you can see the microglial cells, as well as the blood coming out here. The microglial cells are migrating to that location to try to clean up and remove that tissue from the site. So again, I'll just show you right before the stroke that we produced here. There's the stroke, and then you can see that massive movement of the microglial cells to try to remove that dead and dying tissue from that area to prevent any further damage. Again, I'll post this in Blackboard so you can look at it on your computer. I'm not sure how well it'll show up here in Blackboard Collaborate. But the microglial cells play a real important role in trying to, if there's damage or problems with neurons, trying to migrate there and removing that dead and dying tissue. Now, another important glial cell is the oligodendral glial cell, which is fun to say. So why don't you take a moment here to say oligodendral glial cell. What their function is to provide the myelination for neurons. Now, a neuron, axon, has a process that comes off of it that will synapse with other neurons. And the oligodendral glial cell wraps around that axon in a fatty substance. And that aids in the communication of the signal. If you didn't have that there, and we're going to talk more about this in our next section, talking about action potentials and resting potentials. But if you didn't have that fatty substance there, just wrapping around those axons, the neuron would never be able to communicate a signal across that axon. So it depends on that insulation, keeping it insulated from other neurons and the other tissue that's there in the central nervous system. So it's insulated and it helps that signal to continue on. Oligodendral glial cells also serve to prevent random growth in the central nervous system. So they create what we refer to as a non-permissive environment. There are proteins on these glial cells that cause neurons to collapse. So it keeps all the growth of the neurons in check. It suppresses growth in the central nervous system. oligodendroglial cells are only found in the brain and in the spinal cord. So when we're talking about myelination, and this picture up here that's showing, again, another interesting dye, shows you all of the myelination for this oligodendroglial cell. That light green is that myelination, whereas that orange color is the cell body. We can see that's a lot of surface area, and it can wrap around multiple locations across the axon, as we can see here. So we have two glial cells, the oligodendroglial cells, wrapping around those locations, and they can wrap around other axons as well. So they support the communication of information across the axon in the brain and the spinal cord. Well, we know that in the peripheral nervous system there are axons. What supports the communication of information across there or insulates the axons? Those are the Schwann cells. Schwann cells are interesting in that they only wrap around one segment of the axon. So here is a Schwann cell here, here's a Schwann cell here, all the way down it's just one Schwann cell. So they're only providing myelination for a very short segment of that axon. They're very different, and they're only found in the peripheral nervous system. Now, let's say that you're working in the kitchen one day and you're chopping up some vegetables and you sever or you puncture your finger and you happen to cut one of these peripheral nerve cells. Well, what's going to happen when you cut that is that everything after the cut away from the cell body will die. They'll degenerate. But what's interesting is that the Schwann cells will actually start to divide. And as they divide, these axons here, because the Schwann cells don't create a non-permissive environment like the oligodendroglial cells, they actually provide a chemical growth signals to say, grow in this direction. So the axons of these peripheral nerve cells will start sprouting. They're looking for where they need to go. And these Schwann cells, as they're divided, provide both a physical route, an area to grow through, and they'll provide chemical signals to say, grow in this direction. And as it starts to grow through, then the Schwann cells will start to wrap around that segment of the axon. So here we can see that they're wrapping around that segment of the axon, providing myelination. And after about 6 months or so, the neuron will have grown back and reinnervated that location that we had removed that axon from. So Schwann cells are very important in terms of being able to support regrowth in the peripheral nervous system. In contrast, the oligodendroglial cells, they actively inhibit growth in the central nervous system, spinal cord, and brain, but they also don't provide chemical signals or a physical route. So this represents a significant barrier for us in trying to develop treatments or interventions that are going to target the central nervous system. We can see that there's a tremendous amount of plasticity or potential for plasticity in the peripheral nervous system that come from the Schwann cells. It's just these oligodendroglial cells prevented in the central nervous system. So when we're looking to try to treat people that have damage, let's say spinal cord damage or stroke, the oligodendroglial cells really are part of the problem, in that they actively suppress growth and they don't provide chemical avenues for them to grow through. So oligodendroglial cells are one of the challenges for regrowth in the central nervous system after spinal cord injury or stroke. But also astrocytes create a problem because in their attempts to quarantine or block off a part of it, the central nervous system, which is important, if you didn't do that, neurons would continue to die off. They would continue to die, release all their neurotransmitters. The next neuron would become excited and it would die, and it would create this domino effect. Astrocytes create a physical barrier to stop that. But in creating that barrier, they create this scar tissue that doesn't allow those neurons to grow through that. So after spinal cord injury, we can see that we can actually get some growth. There are chemicals, compounds that we can get some growth. This is a spinal cord. This is where the damage is located. And although we can give compounds that suppress the signal from the oligodendroglial cells, we still have that scar tissue that's preventing the growth through that. And so this is a challenge that we're still trying to struggle with, understand, to overcome, to treat people with spinal cord injury, or the same thing with stroke. If somebody has a stroke, dead and dying tissue, you're going to have the oligodendroglial cells that are actively inhibiting the plasticity in the central nervous system. They are also not providing chemical landmarks to grow towards, and the astrocytes are creating scar tissue that makes it difficult, if we can get those oligodendroglial cells to turn off that signal, for neurons to be able to grow through that location. So there's still some challenges, and these are things that people are actively researching to try to develop better treatments. When we talk about our developmental chapter later in this semester, we're going to come back to this idea of trying to understand develop treatments or therapies based on how we build a central nervous system and being able to turn off and on these signals. So now we can turn our attention away from the glial cells and talk more about the anatomy of the neuron, how it collects information, how it functions, somewhat like a factory, some of those characteristics, and talk a little bit about the genetics of the central nervous system. So when we talk about the neuron, we can see that it can be broken up roughly into the dendrites that collect information. So this is a little branch coming in from another neuron, synapsing here on the dendrites. These little bumps here, which you can kind of see in this picture. This is a Golgi stain that we kind of talked about, that Golgi developed and then Cajal refined to support his idea of the neural doctrine. So here we can see that there are these little bumps. Those are all areas where we're having some synapses occurring, or places where one neuron is in close contact with another neuron. And we see that then comes down into the cell body. The cell body functions, or soma, functions to integrate information. It's kind of like the big summation sign. It's what allows you to say whether this neuron should become active or not active, what happens at the cell body. Here, from the cell body, you're going to have an axon, and it's hard to see the axon in this picture, but over here in the cartoon drawing, we can see an axon coming off, and then it will come and synapse on another neuron. So dendrites collect information, the soma, or the cell body, integrates that information and decides what it should do with it, and then the axon carries that decision whether to be excited onto another neuron to excite that or potentially inhibit that neuron. So dendrites collect, soma, cell body integrates, axon communicates further down. That's the flow of information across the neuron. So let's talk about some of the structures that support this information processing. First, we can talk about the walls or the membrane of the neuron. So in a factory, you have walls that protect whatever you're making from the environment. You have some room in the factory that's going to hold the blueprints for what you're making. You're going to have some assembly line that's going to be manufacturing what you're making. And then you're going to do some kind of packaging. You need to package it. You just can't let stuff go out the door unpackaged. Think, for example, that this is a toilet paper factory. Well, you're definitely going to want walls because you don't want it raining in on the toilet paper. You're going to have some blueprint for how to make the toilet paper and wind it up appropriately. Some assembly line to actually take the paper and make it into the appropriate size. And then you don't want to just ship it out without being wrapped in plastic, otherwise it could become wet or something during the packaging. And then you have to have some system to communicate that those products, that toilet paper, to different parts of the country that need the toilet paper. And what we'll do is we'll take this idea, this analogy of a factory, and translate it to a neuron to see that many of these same kinds of processes are in fact operating So the cell membrane is the walls. It protects the neuron from different things there in the environment outside of that. It helps maintain a certain charge. It also helps to keep certain proteins within the neuron separate from proteins outside of the neuron. It's formed by a phospholipid bilayer where there is this is a diagram of a phospholipid, and we see there's a head, and then there's these tails. What's interesting is the head of it is hydrophilic. That means the head likes to be next to water. And it has a hydrophobic tail. Right here we can see this tail portion that doesn't like to be next to water. So if you were to have a bunch of these phospholipids together and you were to put them in water, they would automatically align in this structure, where the heads are oriented towards where the water is located and the tails would be oriented to one another. I like to think of this kind of like if you were to take some olive oil and pour in just a little balsamic vinaigrette. And it doesn't just start to diffuse or start to blend in with the oil. It forms this very strong bubble. It makes these little bubbles of balsamic vinegar that's in the olive oil. And that reflects what happens at the cell membrane level, is that there is a certain part of this protein here, this phospholipid, that wants to be close to other phospholipid components, or these tails, and the head likes to be next to water, and so we form this membrane. That's what this part of the neuron is this phospholipid bilayer. And so it's very hard for things to get in and out of here. And we'll see that we've evolved, or there have been these different adaptations to allow us to get information or products in and out of this phospholipid bilayer. So moving in, where is that blueprint room? Where is we keep information of what we should be making or doing within the neuron? That's in the nucleus. The nucleus has all of our chromosomes. And our chromosomes are basically, I would say, pretty similar to a book of blueprints. And within each book of blueprints, we're going to have a certain number of products that we can make. So we have a certain page on a chromosome is a gene, and that is a certain product that we can make. So a gene tells us to make this protein, which could be used to make something that the neuron needs to use, such as maybe a neurotransmitter or some other item that the neuron needs to live. So again, a chromosome, which we have 23 pairs of chromosomes. Each pair is within each cell of in the body, and within the central nervous system we have 23 chromosomes. Here is a single chromosome. If we were to pull that off, there's a little segment there that's going to be coding for some product we want to make, and that's going to be a gene. A gene is composed of DNA, and the DNA is kind of like the words on the page. The gene is like a page of the blueprint book, whereas a chromosome is the blueprint book itself. And the nucleus is the blueprint room that houses all the books with all the blueprints of how to make anything. So when we look at this, what we see is the nucleus houses all the chromosomes. A single chromosome is kind of like a blueprint book, and a single page out of the blueprint book that refers to how to make a certain kind of product. Maybe it's something the neuron needs, like a neurotransmitter. That gene will be expressed in a certain region on that chromosome, and that gene then is composed of DNA, this double helix stranded molecule here, where we have different base pairs that combine to make up that gene. So again, you can review this in the book. I know this may be a little bit much. That's why I'm trying to go slow. Remember, you can replay this to step through this a couple of times. Now, after we have the gene segment. Remember, this builds something that the neuron needs, and we're going to tell you what that is in a little bit, what genes code for, and give you some examples of what happens if there's a mutation or there's something wrong with genes. A gene is a certain segment of the chromosome. And let's say that there's a call that we need to make something more for the neuron. When we do that, what happens is that segment of the gene can actually unwind. And when it unwinds, we start to make a messenger RNA. Basically, messenger RNA is kind of like a photocopy, or we're transcribing what is coded for in that gene. So what happens is that gene will unwind, messenger RNA will be transcribed from those genes, and then that messenger RNA will then exit the nucleus. So that exiting the nucleus will then go out to help make that what's necessary. I kind of think of this as, think of a factory, and we have those blueprint books, and we don't necessarily want to take the blueprint books out of the blueprint room, because if we do, they'll start to get all dirty, people will spill coffee on them, somebody will drop part of their donut and close the book, and so we don't want to damage them. So the best way to do that is to photocopy them or transcribe notes, transcription, the code that we have in our gene into messenger RNA, then that can go out to the factory floor, and that's where we'll then use that to make the product that we need for the cell. So after the messenger RNA has left the nucleus, it'll go out to the endoplasmic reticulum, where there are plenty of ribosomes. Remember, maybe in biology class in high school, you learned about the endoplasmic reticulum. They're smooth and rough. The rough endoplasmic reticulum has a bunch of ribosomes, and that's where the messenger RNA will go and interact with the ribosome to make the proteins through a process of translation. So it takes the code that the messenger RNA is carrying and turns it into the protein that actually will be used by the cell or the neuron to increase or enhance the functioning. So the process involves, first you'll have a gene unravel, and then the DNA will be copied to messenger RNA through a process of transcription. From the messenger RNA leaving the nucleus, it will go out to the endoplasmic reticulum where the ribosomes are located. Once the messenger RNA interacts with the ribosome, you'll translate that into proteins through a process called translation. Now, what are these proteins? Well, remember, each little component there of a protein is an amino acid, and we can build them together to make a protein. Proteins can vary in structure and complexity. So we can have very simple amino acid chains to where they can become pleated sheets, to where they can become helixes or a tertiary or a quaternary structure. And what are these? What are we talking about that the neuron needs or other cells in the body need these proteins for? And there are an example of what these proteins could be are channels. So remember, we talked about the phospholipid bilayer, that is the cell membrane, and it doesn't allow a lot of movement of chemicals through. Well, we can have a channel that specifically allows, for example, potassium to flow through the cell membrane. So it's like a little window that's always open that allows potassium to flow through. So you can have channels, and those are some of these quaternary structures that are used to allow certain ions to flow through the phospholipid bilayer. You could also have gates. Gates are just a step up from channels, and that something has to happen to open the gate, and once the gate's open, then certain ions or chemicals can flow through the membrane. So here we have sodium, and normally the sodium can't get through. However, if the gate opens, now all of a sudden sodium can pass through this membrane. So there has to be something to happen to allow sodium to pass through that gate. If that doesn't open up, then sodium can't pass through. Finally, we can have pumps. Pumps work to take certain ions or chemicals that are on one side of the neuron and move them to the other side. So we can be bringing sodium in or taking sodium out, putting potassium in, putting potassium out. So we can have this continuous exchange. And this is a protein that can be made from expression of the DNA process of transcription to translation. And then they can turn into channels to allow certain chemicals to move across the membrane, gates that have some conditions that need to be met in order to allow certain chemicals to move across the membrane, or pumps that sit there and actively transport certain chemicals across the membrane, kicking sodium out and bringing potassium in. So just to review one more time, a gene represents a certain segment on a chromosome that codes for a certain protein. We don't want to take the genes out of the nucleus, they may become damaged, so we transcribe them into messenger RNA. The messenger RNA leaves the nucleus and then goes out to the endoplasmic reticulum where there are ribosomes. Those ribosomes then take that messenger RNA and turn it into proteins through the process of translation. Those proteins can vary in structure and complexity to where we can get quaternary structures of those proteins. Those proteins could be channels that allow ions to move across the phospholipid bilayer. They could be gates that have some conditional rules when those ions can move through, or they can be pumps where they actively move them across the membrane to serve some purpose. So after we've built our proteins by the endoplasmic reticulum, what we need to do is package them and send them where they're needed in the neuron, and insert them into the membrane, for example. The Golgi bodies, also discovered by Golgi, hence the name, actively will wrap up those proteins into vesicles, and then those vesicles will become latched on microtubules. Microtubules are kind of like railroad tracks that serve as a transport system for these proteins. Here what we see are examples of vesicles that have some protein in them, or some vesicles that have the protein inserted into the membrane of the vesicle. And as they move across here, some will be inserted into the membrane of the axon. Some will be released directly into the environment. So there's a lot of variability in which those proteins can be moved around the neuron. Damage to these microtubules can result in kind of crumpling the tracks. And what we'll see when we'll talk about Alzheimer's disease, that those neurofibrillary tangles represent some of these systems to move proteins across the neuron becoming compromised, and this neuron will no longer be able to move the proteins around like it needs to, and the cell will die. And what will be left will be these neurofibrillary tangles. We'll talk more about that later on. It's just an example of the importance of the Golgi bodies and the microtubules for the health of the neuron. If you disrupt that, the neuron will die. So let's talk a little bit more about the genes that are within the nucleus and this idea of what an allele is. And maybe you've heard this term before, maybe it's not quite familiar. An allele is a gene, it's a version of a gene, and you could have a normal allele or you could have a mutated or a different allele. And in Tay-Sachs disease, what we see is that it's a recessive gene disorder, a recessive allele disorder. Tay-Sachs disease involves a dysfunctional protein, and it fails to break down lipids, fats, within the central nervous system. This usually is observed very early in the development of a child. So almost from birth, you'll start to see some seizures in the child. And the child will be blind. Typically, there will be a degenerative motor disorder, and there will be mental delays, and usually the child will die within the first few years with Tay-Sachs disease. And it's because of this recessive allele that produces a dysfunctional protein. This is also in your book if you want to review it there. With this, what we see is that we could have a normal carrier and a person that doesn't have the allele, the hex A allele for that. Remember, everybody has 23 pairs of genes, and you could have one of those pairs that has the normal allele and one that has the hex A allele. In this condition, where it's a normal carrier and just a normal person, we'll see that no child will have Tay-Sachs disease in this. We'll have a 50-50 chance of being a normal carrier or a normal individual that doesn't carry that gene. This is different than if two people have that are normal carriers. If they have that allele, they're the carrier for the hex A allele, then what happens is that they have a 25% chance that their child will develop Tay-Sachs disease, where they'll have 25% that their child will be normal, and there's 50% chance that their child will be a carrier for that. Whereas if their child is from a normal individual and a normal carrier, they will not develop Tay-Sachs, but there's a 50% chance of becoming a normal carrier. So you can see how an allele like this could continue on in society, because there is a chance where there are some normal carriers and there's only a 25% chance of developing Tay-Sachs if two normal carriers have children. So again, this is an example of a recessive allele disorder that involves that protein. And the problem is that this dysfunctional protein that's produced by this allele fails to break down fats in the central nervous system. Now, we can also look at dominant allele disorders, such as Huntington's disease. Huntington's disease involves, if you have just one of these alleles, then you're going to code for a protein that kills brain cells in the basal ganglia and cortex. But because this allele takes a while to make these proteins, and these proteins take a while to accumulate, people don't start showing signs of Huntington's disease until their early 40s, well into that reproductive age, where somebody could have a child and not be aware that they're going to develop Huntington's disease. Again, the Huntington allele codes for a protein that as it accumulates, will kill brain cells in the basal ganglia and cortex. Now let's look at the pattern of genetics for the individuals with either a normal person or a carrier for the Huntington allele. If a carrier was to have a child with a normal person, we see that you have a 50-50 chance of being normal and a 50-50 chance of developing Huntington's disease. Now, if two carriers have a child, we see that you have a 75% chance of developing Huntington's disease or inheriting that Huntington allele and developing Huntington's disease, whereas you have a 25% chance of developing or inheriting the two normal alleles and not developing Huntington's disease. So a very different pattern when we look at dominant versus recessive alleles in these diseases. Well, it's clear that there are certain factors that influence the genes and the expression of proteins, as we can see from both Tay-Sachs disease and Huntington's disease. Your book goes into some detail talking about Mendelian genetics and some of the early work looking at peas and pea flowers and discovering this kind of relationship here. This was a discussion of how these proteins can translate to certain disorders that influence the central nervous system. Tay-Sachs, again, causes seizures, whereas Huntington's disease causes death in certain neurons in the basal ganglia and causing motor disorders. Now what we want to turn to is this idea of phenotype plasticity. And this is really getting into the idea of epigenetics, this idea that there can be changes in the DNA that can influence the expression or the phenotype of the individual. And so you can change the DNA and cause some interesting changes in the DNA that may be translated across generations. And we'll talk about 3 ways that we can change the expression of the DNA and then give you a piece of evidence that this can change across generations. And we'll talk about that now. So one mechanism is the fact that DNA is normally pretty tightly wound around histones. And so here we can see this is DNA molecules wrapped around these histones. And normally the DNA, when it needs to be unraveled for a gene to allow for transcription, it can unwind and allow that to happen. Well, what we can have is certain environmental conditions can cause these histones to be bound to methyl groups, or CH3, these little red units here. And when it becomes unwound, or when it becomes bound, that means that segment of the DNA is blocked and it can no longer be available to transcribe for those proteins. So we've essentially turned off the production of proteins there. So that's one way we can prevent the DNA from becoming unwound to those histones. Also, we know that once it becomes unwound from those histones, the DNA has to come apart for the messenger RNA to be built. Well, the methyl group can also attach directly to the DNA and prevent it from unwinding. So that's eliminating those base pairs from coming apart and producing the messenger RNA. So we see here that when that methyl group attaches to the DNA, now we block transcription again, turning off those proteins. So we might not make any more of those channels, or any more of those gates, or any more of those pumps. And finally, we can have certain processes where the methyl group will bind to messenger RNA, and that will prevent the transcription, or taking that messenger RNA and turning it into proteins. So these are 3 different ways in which we can change the expression of the proteins. And these could be a mechanism to change, have a behavioral experience in one generation and have it translate to another generation. So what we'll talk about last here in this section is an interesting study, Norrbotten, Sweden study, looking at individuals that would go through either feast or famine. So they were able to go through and categorize individuals from this region. Their grandparents and great-grandparents had either gone through a feast where there was plenty of food, or a famine where food was very scarce, and follow their grandchildren and their children to see, was there any kind of effect of going through a feast or a famine for their health consequences. And what they found is that if they experienced a feast or a famine the year before they entered puberty, so around 8 to 12 years of age, they found that their children and their grandchildren, if they experienced a feast, they had higher cardiovascular problems, higher instances of diabetes, and shorter life. So if their grandparents or parents in 1801, 1822, or 1828 were experienced a feast between the ages of 8 and 12 in 1801, 1822, and 1828, they had poor health outcomes. Whereas if there was a famine in 1801, 1812, or 1821, they tended to have better health outcomes, where you'd see that they would have better cardiovascular function, fewer instances of diabetes, and a longer life. So feast, the grandparents, if they went through a feast, they tended to have poor health outcomes, whereas if they went through prior to that onset of puberty, if they went through famine, their children and their grandchildren tended to have better health outcomes. What makes this interesting and really suggesting epigenetics, that this influence could be translated, this behavioral experience could be translated, is that it's completely sex-linked. This relationship only occurred if it was a male that experienced feast or famine, and it gave birth to a male child, and they had a male grandchild. So it was only in those sex chromosomes was this passed through. If it was a male that gave birth to a female and then had a granddaughter, that relationship wasn't observed. If it was a female that went through feast or famine and their child was a female, then they showed it, or their grandchild was a female, showed it. But if a female had a male child or had a male grandchild, after that they had to have that carried through, then they showed that similar link. But it did not show if there was a mismatch, where a female went through feast or famine, had a male, then that male would not show it. If that male had a female, that wouldn't show it. So you had to have that continuous line suggesting a gene, a change in the way those genes were expressed because of that early experience from the 8 to 12 years of feast or famine. So this is a really intriguing result, suggesting that these experiences across life can dramatically influence not only one's behavior and the way the proteins are expressed, but could have consequences for multiple generations. And when we talk about development, we'll see that there's a lot of effects that environment can have on there, and people are getting very excited about this idea of epigenetics, understanding it, and maybe trying to find ways to manipulate this. So this concludes our discussion of the functional units of the nervous system. We talked about neurons, we talked about glial cells, their roles, and some of their anatomy. We focused in on the neuron to talk about some of the anatomy, and we talked about this idea of how genes are important, and you can have these different types of mutation, and how epigenetics, or this phenotypic plasticity, can change within a generation and potentially between generations. So again, I hope you're taking notes on this, taking notes in the book. Remember to then take the lecture quiz. You only get one shot at those. Take the practice exam. You get 3 shots at those. Both are open book, open note, as much as you can. And then since this was our Chapter 3, as long as you feel like you really understand the material and you did well on those 2 indexes, the practice and lecture quizzes, you're now able to take exam 1. So consider that in terms of where you're at. The exam 1 covers origins, it covers neuroanatomy, and it covers this chapter, functional units of the central nervous system. And there will be 30 questions that will be multiple choice, as well as 2 short answer questions, much like those lecture quiz questions. So make sure you review those, really study, because this is worth the exam 3 is worth 20% of your grade. So thank you, and I look forward to sharing with you information for our next unit where we get to talk about neuronal communication. So now this will provide further information building on what we've talked about, how neurons communicate with one another. Talk to you soon. Bye.